So we're going to talk about a few other tests, of course, other than the t-test in this unit. We'll talk about the chi-square test, we'll talk about the Wilkinson rank test. But first, we're going to focus on an ANOVA test. Because what we've done so far with the t-test is we compared two populations. But in reality, we often have more than two populations here. So when we do that, we rely on another type of test, which is called the analysis of variance analysis test, which is also known as the ANOVA. So this is a bit of a cheat sheet of what you can do. So we're talking about normal distribution, normally distributed data. And these are the goals. So if we want to compare one group to a hypothetical value, the example of is the bread really different to 580 grams, then we can conduct a one-sample t-test. In R, we would do it using the t-test function. We can compare two paired groups. We spoke about pairing. Again, the t-test function will be pair equals true. I mean, like you'll see it when you get to it. You can compare two unpaired groups. Again, a two-sample t-test. Now we're talking about comparing more than two groups. And for that, we're going to need an analysis of variance, or in R, the AOV. There are tests that mimic the test for normally distributed data that are non-parametric, that are not relying on the parameters of the normal distribution. And that's what we use when our data is not normally distributed. And we also have tests for data that's not continuous. So for example, if we have ratios of binomial data, yes, no, then we use other tests. Okay, we're going to get to those later. So we're still dealing with tests that fit normally distributed continuous data. And now we'll talk a little bit about the analysis of variance. So if previously I had two groups and their means, and I wanted to know if I take this point here, does it belong to this mean? Does it belong to that mean? Or can I not tell them apart? Can I not place this spot here in group 1 or group 2 with confidence? Now we have a situation where actually there are 3 groups or more. Okay? So I can have an experimental treatment. I can grow some plants, right? And I can have 3 groups. I can have plants that are grown at elevated CO2 conditions. I want to know how elevated CO2 is impacting plant water use, okay? Because it affects the water cycle. I have some plants growing at elevated CO2 concentrations. I have some plants that are growing at ambient CO2 concentration. This time I have 2 groups, t-test, right? What if I have another group, which is plants growing at pre-industrial CO2 concentrations? Because right now we're at 400. We started at 280 not that long ago. Okay, so if I'm going to have something at 800 ppm, something at 400 ppm, something at 280 ppm, I'm already comparing more than 2 groups. This is not the task for a t-test. This is the task for an ANOVA. So when I compare more than 2 groups, my hypothesis becomes a bit tricky, where I have what we call the omnibus null hypothesis, which is my null hypothesis is that all the means are the same. Okay, so the mean of population 1 is equal to the mean of population 2, is equal to the mean of population 3, is equal to the mean of population 4, 5, 6. You know, so actually, if I look at the water use of population 1, of the pre-industrial, of the ambient CO2 concentration, of the future CO2 concentration, I can't tell them apart. Okay? So that's my null hypothesis. And then I have an alternative, and the alternative is tricky because it doesn't actually give me the answer I want per se. It tells me at least one population mean is different from at least one other. Okay, so what I can get is that the ANOVA will show a significant difference, but I don't actually know why. Is it the pre-industrial is different to the future? It will, or is it the ambient different to the pre-industrial? Yeah, you know what I mean? It's enough that one group is different to one other that I will get a significant difference in the ANOVA tests, but I will have to conduct other tests later, what we call post hoc tests, to work out who's actually different to whom. Okay, so the omnibus is all the means are different, the alternative is at least one is different. Or in other words, not so. Okay, so all the means are different, all the means are the same, alternative, not so. Okay. So why not just use the t-test for, I'm going to look at the difference between population 1 and 2, 1 and 3, 2 and 3, right? I mean, I could do that, right? I could do a series of t-tests and work out who's different to whom. Why am I using an ANOVA, right? So first of all, there's an issue of, well, if you have a lot of groups, you're going to have to do, you're going to have a lot of pairs that you need to test. And so as the number of groups grow, the number of paired comparison grows quickly, okay? Much quicker than the number of groups. Right? So what I'm getting at is every time we say there's a difference, we are taking a risk that we're wrong, right? We have that 5% chance that actually I'm saying they're different, but they're not, right? As I repeat the test again and again and again on this grouping, on 1 and 2, on 1 and 3, on 1 and... Eventually, I'm going to hit the point where I got it wrong, that I guessed wrong. I'm taking too much risk here. I'm adding up those 5% every step of the way. Okay, so what I want is a single test statistic that will be tested, not to repeat t-tests on all possible pairs. So my analysis of variance, ANOVA, as it says on the box, uses the variances to determine whether the means are different. So by that I'm saying, I have eyes, right? Or I have a face, and you have a face, and a cow has a face, okay? And I can tell apart my face from the face of a cow, which is a good thing, right? But why can I do that? It's because my face and your face is not the same. There's a variation in human facial features, right? There's also a variation between cows, huge variation. But the variation between my face and a cow's face is just so much greater than the variation within the human population or the variation within the cow's population. So the between group variation becomes really important in the ANOVA. If I think about, I have school-age kids, and my son is 9 today. And when you look at a group of 9-year-olds, they have a huge variation in their height. Okay? So a lot of them are are still kind of little. Some of them have shot up already and are really tall. If you look at a group of 15-year-olds, they're already easier to identify because the variation in the population is smaller and the variation between the population. So if I just pick up a random kid at school, and I don't suggest you do that, I could probably say this is a year 6 kid, but I probably can't say this is a year 2 or a year 3 kid because the variation inside that population is just too big to be able to identify one kid to a population. So I have the between group variation, okay, the variation between group 1 and group 2, and I have the within group variation of within group 1 and within group 2. Okay, so by comparing the variances between group means versus the variance within groups, we can determine whether the groups are all part of one larger population or separate populations with different characteristics. So this time we're looking at the characteristics of the distribution, really, and we're working out, is this a different distribution to that? Is the variation different? This time, instead of calculating a T statistic, we calculate something called the F statistic. And the F has a PDF of its own, again, dependent on the number of samples on the side of the population. Okay? So we have the F is pretty much, and we'll see in a minute, but it's pretty much something about the variance over all the samples and the variance within the groups. So we have different, these are the number of groups we have and the number of samples we have, okay? And that's the two things that determine the shape of the distribution. So we'll need to know there's two things when we calculate the F statistic and its p-value. I'm going to give you an example for an ANOVA test. Here are different soil horizons. We have an A1 horizon and A2 horizon. Those of you who are taking soil science with Vanessa would know all about this. We have some core stones at the bottom of the soil core, and this is the soil profile, and it has different horizons, and these different horizons vary. I mean, like you can see from the color, they vary in their structure, in their chemical composition, and in all those microbial pathways that occur in them. And I can take these soil cores, right, from different parts of, in this case, she went to different sites, RMC, SC, TAC. This is a proper spreadsheet. I actually don't know where these places are. There are 3 sites here, and she's collected the soil cores from the back swamp, which is probably the part where the swamp kind of goes back to a dry landscape. And these are the horizons that she sampled, so only the A horizon, so that's this bit here. And these are replicates, how many times she sampled. Okay, so she sampled in different sections of the swamp. She took 3 cores from each site. EC is the salinity, so we have a continuous variable here, and it was very uniform in this swamp, much less in this swamp and much less in this swamp. It has EH, which is the redox potential, or which suggests well-aerated soils over here. These are redox potentials that suggest a lot of oxygen in the soil. And this is the pH of the soil, which is actually quite acidic. Okay? And I want to know the soil acidity vary among the 3 sampling sites. So my null hypothesis is that, oh no. And my alternative is, yes, it varies across the 3 sites. I don't know which site, okay, I just know that it varies. So why do I need an ANOVA? Because I have 3 sites. I have 3 populations I'm comparing, okay, 3 populations, 3 in each population, that's not ideal, right? And I'm conducting what's called a one-way ANOVA. There's only one thing varying here, which is the site. Everything else, the same. And I can plot the different sites, the pH at the different sites. And what you can see is the means are really different. There's very little variation around the mean. So all the this box plot that shows the variation around the mean is very condensed. And I can take an F, calculate an F. So if I have an F that's equal 1, right, these are the same. The variation between the groups is the same as the variation within the group. So I can't tell them apart. If my F is bigger than 1, the difference between the groups, the variation between the groups is greater than the variation within the group, indicating the groups are different to each other. Now, using R, I can build an ANOVA. Okay, so remember we can use the equal sign, almost the same as we can use that arrow sign. So if I have an arrow somewhere, I'll show you next time. And I will do an AOV in ANOVA of pH, and this little squiggly line, the tilde, is as a function of site in the dataset soil. Right? So if I go pH as a function of site, and I ask for a summary of this ANOVA, what I'll see is the site has 2 degrees of freedom. Why? Because we have 3 sites, we take 1 away. The sum of squares, the mean sum of squares, so that's the top, the bottom, My F value is humongous, okay, way bigger than 1. The probability that my populations are the same is very, very small. So my F is very big. The chance of getting an F bigger than this one is basically 0. So I'm going to reject my null hypothesis. pH is not the same on one side. We don't know which side is different, though. I can do a post-hoc analysis to separate the tests later. So next lecture, we'll talk about the chi-square test. But that was the one-way ANOVA, the one-way analysis of variance. It's just the beginning of ANOVA. We're going to practice them in the